He's got a question. Go
Yeah,
ahead.
sure. Can you give us a couple of minutes on optogenetics and how that might fit into this, or not fit into this?
Sure. Yeah. So that's kind of another side of the story. So what I was talking about, here at the end was mapping the structure, and there's also the issue of mapping the functionality and the dynamic patterns. Optogenetics is kind of the opposite. Is if you want to do a very pinpoint intervention, and you want to turn on a specific type of cell at a very specific moment, then you can do that by shining light, and then that activates that neuron, or that right now it's a kind of a set of neurons that are expressing a certain protein. So this is really powerful as kind of like this pinpoint control mechanism. But then the question is, well, what do you want to really be controlling, right? So there are these hundreds of different types of neurons in these very complex circuitry. There's a lot of possible experiments, perturbation experiments that you could do, and it's sort of very unconstrained, I think, without having a map, right? So I could stimulate the thalamus and see what happens with the cortex, and there's lots of things you can try. But I think we also need this just raw structural map. But they're two sides of the same coin, right? Because eventually what you would want is like a map that's being done in the context of very targeted stimulations, where you see one cell turns on, and how does that propagate a wave to the rest of the cells. And so.
Okay, Peter's got a question.
I was recently at this pretty amazing DARPA event.
I was there too. Yeah.
Oh, were you there too?
Yeah.
So, great. Then you'll know exactly what I'm talking about. There's an experiment where they claim to have shown or imaged a quote-unquote mouse thought. Whatever a mouse thought is, I don't know. I'm not a neuroscientist, by the way, but I studied with Michael R.B.
Cool, yeah.
But super impressive experiment.
Yeah.
But is that really a mouse thought?
I think it's at best a piece. So the part I was talking about, just because you have only 15 minutes, right? I was talking about the structural molecular aspect. I mean, the other aspect is mapping the dynamic patterns, of course. And people are right now up to a few thousand neurons using the technology that they were excited about at the DARPA conference, which is also optical imaging, look with a microscope. And this is exciting, and people at the DARPA conference are talking about, can we actually do this in humans, right, and look at neural populations. It's very interesting that they're thinking about this.
Is it really a thought?
It's pretty far out. Well, I think, if you took even a single cubic millimeter of tissue, that's 100,000 neurons, right? So we're still only seeing a tiny subset of just what would be in a single cubic millimeter, right? I think the thought really probably has to do with different, like I was showing a diagram, different parts of cortex interacting and then making a decision. That's not something you can see just by looking at some really local area. So I think we need to eventually image the whole brain potentially in order to actually see a thought, or to see the physical manifestation of a thought, which would then have to decode what that means. But
How about one more question? Anybody's got a question?